Hi everyone, this is the first lecture in Chapter 8. Chapter 8 is on microbial genetics and genetic engineering, and this first mini lecture is going to cover DNA structure and DNA replication. So when we talk about DNA, there's a code that we think of which is made up of nucleotides. These are the basic unit of a DNA structure. A nucleotide can be broken down into a phosphate group, deoxyribose sugar, and a nitrogenous base. Nucleotides are covalently bonded to each other, and it's the sugar phosphate linkage that becomes this backbone of these strands of DNA. Each sugar will attach in a repetitive pattern to two phosphates. And so one of them attaches at the 5' carbon, and one is the 3' carbon. And what you'll get is the two strands, they'll have the 5' and 3' opposite each other to make a complementary strand. So the nitrogenous bases attach along the strand by covalent bonds, and we have purines and pyrimidines that join with complementary bases. So you'll have ones that pair, and it's always the same rule. You can see here, adenine or A always will pair with thymine T, and guanine, which is G, always pairs with cytosine. So no matter what, if you see adenine, you always know that on the other side, the complementary strand, it will be thymine. And this bonding of these purines and pyrimidines across each other with this weak hydrogen bond allows for this molecule to be easily unzipped, is the best way to describe it, which allows perhaps mRNA to be formed off of it, or a new DNA strand from the information encoded in these bases. So when we look at the actual structure, this is showing three different views. Mostly we see this kind of double helix is what you would see. So you can see this red strand at the top, it starts with the 3', and at the bottom that red strand has the 5', but its complementary strand has the 3' down here and the 5' up here, and this allows it to then form this kind of ladder or helix. This is showing a little bit more complex pattern of that with the hydrogen bonding between the purine and the pyrimidine. These are just different ways of looking at it. This is a figure in your book, so you can take a look at this and spend a little bit more time on it. But you can see here, the green here is guanine. It's showing it paired with the C. Of cytosine, you'll see that these pairs always match up, A to T, C to G. So this is a great view of that DNA structure. When we talk about this double helix, like I said, this is that arrangement of one starts at the 5' to the 3', and then the other one has that opposite direction. There's a term for this. This is called anti-parallel arrangement. Also, the order of these bonds between the carbon and the deoxyribose helps keep track of the direction of the two sides. So you know that one's running from 5' to 3 and the other is doing the opposite. And this is a really important factor both in when we look at DNA synthesis, so when we are replicating DNA, and also in protein production, which we'll get into a little bit later in this chapter. So what is so important about this, or complex about this genetic language that we have in DNA? Because we're talking about only 4 nitrogen bases, right? We have just A, T, C, and G. So the idea that that's very complex might seem counterintuitive. But when we look or consider that a segment of DNA can have 1,000 nucleotides, what we're really looking at then is 4 to the power of 1,000 different sequences that are possible. So you can see that this actually is a huge variation that we can see even though it's only 4 bases. And that's one of the kind of amazing things about DNA. When we talk about DNA replication, so we have a strand of DNA and we're wanting to replicate it, one of the things to always keep in mind in DNA replication is that it is semi-conservative replication. And what semi-conservative means is that as we unwind or unzip this DNA, when we replicate one side of it, that will actually be a new whole DNA. So it should have one strand that is the original, and then the complementary strand, which is the new strand. And that would also be true about the other strand of the original that we unwound. So it's a bunch of different enzymes that come in to do this. They'll separate that strand of existing DNA. They'll copy one strand and produce two complete daughter molecules. And like I said before, each daughter molecule, although it's identical to the original, one side of it will be new and one side of it will be old, or the actual template. So that's what it means by semi-conservative replication. And then when we talk about DNA, there are a lot of details that come into DNA replication. Most of what happens with DNA replication is carried out by the enzyme DNA polymerase 3. However, it is limited by the fact that it is unable to begin, so to actually start making a chain of nucleotides. It just has the ability to actually add nucleotides to an already existing chain. So it's really the elongation portion that DNA polymerase 3 does. And it can also only add nucleotides in the 5' to 3' direction, which means it can really only do it on one side of the DNA chain. So down below I've put the table from chapter 8, it's 8.1, and this is just going over some of the enzymes that are involved in DNA replication and their functions. I'm not spending a lot of time on this because this should be review for you. If this is difficult though and you need some extra help, please let me know. So the main ones here are helicase. This is the one that's responsible for unzipping the DNA helix. We have primase, and primase is the one that will actually synthesize an RNA primer, which then the DNA polymerase 3 can build off of. So we start out with this piece of RNA, and then we add new bases to form a DNA chain. We then will have DNA polymerase 1 come in and remove the primer. It will also close any gaps that may have been missed in the chain and repair any mismatches. And then ligase can come in and bind any places where there may have been repair. And then our last ones that would come in are the topoisomers and these isomerases, and these are 1 and 2, and they do both supercoiling, so coiling at the end, or untangling when we're beginning. This is a great table for you to look at. This goes through each step-by-step part of DNA replication. If this does not seem like review to you, this is where I would start. It goes through each step. In addition, it will tell you which enzyme is needed. So please take a look at this, and then if it's still confusing, you certainly can contact me. So the elongation of that DNA complementary strand, and then what happens to terminate it and make those daughter molecules. So in bacteria, some of them can add nucleotides super fast. This is 750 bases per second at each fork. So you can imagine how fast it can do this elongation or making the DNA replication. So like I said before, the DNA polymerase 1 will remove that RNA primer, that starter of our DNA, and it will replace it with DNA. And then the ligase will move along the lagging strand and link these fragments so that you have that complete synthesis and separation of two daughter molecules. Now, mistakes do happen, but it's actually very rare. It's only approximately in 10 to the 8th to 10 to the 9th bases, so that's very few mistakes when you think about the size. And most of them would be corrected by DNA polymerase 3. It would actually come through and edit that DNA strand. And then lastly, kind of that completion of this, what would happen. So this is a little bit different than eukaryote cells. So if you're remembering from biology and remembering how this looks, normally we'd see that nice long helix in a kind of a straight line. We're looking at this circular chromosome in a bacteria. You can see there's a fork formed here and it's starting to replicate, and a fork formed here. And what ends up happening is when we have two complete circles, there will actually be a break here and separated out to two daughter molecules. The red strands are representing the original strands, the blue the new, and you can see here that Each of these daughter cells then possesses one strand that is new and one strand that was original, hence that semi-conservative replication. That's the first of our lectures in Chapter 8. If you have any questions, please let me know.